Welcome to Biology, the scientific study of life. Many animals regulate their internal environment within relatively narrow limits. More than a century ago, French physiologist Claude Bernard made the distinction between the external environment surrounding an animal and the internal environment in which the cells of the animal live. The internal environment of vertebrates is called the interstitial fluid. This fluid, which fills the spaces between vertebrate cells, exchanges nutrients and wastes with the blood contained in microscopic vessels called capillaries. Bernard also recognized that many animals tend to maintain relatively constant conditions in their internal environment even when the external environment changes. Pond-dwelling hydra is powerless to affect the temperatures of the fluid that bathes its cells, but the human body can maintain its internal bond at a more or less constant temperatures of about 37 Celsius degree. The human body also can control the pH of the blood and interstitial fluid to within a tenth of a pH unit of 7.4, and it can regulate the amount of sugar in the blood so that it does not fluctuate for long from a concentration of about 90 milligrams of glucose per 100 milliliters of blood. There are times, of course, during the development of an animal when major changes in the internal environment are programmed to occur. For example, the balance of hormones in human blood is altered radically during puberty and pregnancy. Still, the stability of the internal environment is remarkable. Today, Bernard's constant internal milieu is incorporated into the concept of homeostasis, which means steady state or internal balance, one of the main objectives of modern physiology. And the theme of this unit is to study how animals maintain homeostasis. Actually, the internal environment of an animal always fluctuates slightly. Homeostasis is a dynamic state, an interplay between outside factors that tend to change the internal environment and internal control mechanisms that oppose such changes. Regulating and conforming are two extremes in how animals cope with environmental fluctuations. An animal is said to be a regulator for a particular environmental variable if it uses internal control mechanisms that to moderate internal change in the face of external fluctuation. For example, a freshwater fish is able to maintain a stable internal concentration of solutes in its blood and interstitial fluid, even though that concentration is different from the solute concentration of the water it lives in. The fish's anatomy and physiology enable it to moderate internal changes in solute concentration. An animal is said to be a conformer for a particular environmental variable if it allows its internal condition to vary with certain external changes. For example, many marine invertebrates, such as spider crabs of the genus Libania, live in environments where the solute concentration salinity is relatively stable. Unlike freshwater fishes, Libania does not regulate its internal solute concentration but rather conforms to the external environment. Regulating and conforming represent extremes on a continuum, and no organism is a perfect regulator or conformer. Furthermore, an animal may maintain homeostasis while regulating some internal conditions and allowing others to conform to the environment. For example, even though a freshwater fish regulates its internal solute concentration, it allows its internal temperature to conform to the external water temperature. More about mechanisms that animals use in regulating certain aspects of their internal environment. Mechanisms of homeostasis. Mechanisms of homeostasis moderate changes in the internal environment. Any homeostatic control system has three functional components: a receptor, a control center, and an effector. The receptor detects a change in some variable of the animal's internal environment, such as a change in body temperature. The control center processes information it receives from the receptor and directs an appropriate response by the effector. Let's consider a non-living example of how these components interact. The regulation of room temperature, as shown in this figure. In this case, the control center, called a thermostat, also contains the receptor, a thermometer. When room temperature falls below a set point, say 20 Celsius degree, the thermostat switches on the heater, the effector. When the thermometer detects a temperature above a set point, the thermostat switches the heater off. This type of control circuit is called negative feedback because a change in the variable being monitored triggers the control mechanism to counteract further change in the same direction, owing to a time lag between reception and response. The variable drifts slightly above and below the set point, but the fluctuations are moderate. Negative feedback mechanisms prevent small changes from becoming too large. Most homeostatic mechanisms in animals operate on this principle of negative feedback. In fact, your own body temperature is kept close to a set point of 37 Celsius degree by the cooperation of several negative feedback circuits. In contrast, to negative feedback, positive feedback involves a change in some variable that triggers mechanisms that amplify rather than reverse the change during childbirth. For instance, the pressure of the baby's head against respirators near the opening of the uterus stimulates uterine contractions, which cause greater pressure against the uterine opening, heightening the contractions, which causes still greater pressure. Positive feedback brings childbirth to completion. It is important not to overstate the concept of a constant internal environment. In fact, regulated change is essential to normal body functions. In some cases, the changes are cyclic, such as the changes in hormone levels responsible for the menstrual cycle in women. In other cases, a regulated change is a reaction to a challenge to the body. For example, the human body reacts to certain infections by raising the set point for temperature to a slightly higher level, and the resulting fever helps fight the infection. Over the short term, homeostatic mechanisms keep body temperature close to a set point, whatever it is at that particular time. But over the longer term, homeostasis allows regulated change in the body's internal environment. Internal regulation is expensive. Anyone who pays utility bills is aware of the energy costs for heating or cooling a home to maintain a comfortable interior temperature. Similarly, animals use a considerable portion of the energy from the food they eat to maintain favorable internal conditions. Thermoregulation contributes to homeostasis and involves anatomy, physiology, and behavior. We will examine one example of how animal form and function work together in regulating the internal environment, specifically the regulation of body temperature. Thermoregulation is the process by which animals maintain an internal temperature within a tolerable range. This ability is critical to survival because most biochemical and physiological processes are very sensitive to changes in body temperature. The rates of most enzyme mediated reactions increase to threefold for every 10 Celsius degree temperature increase until temperature is high enough to begin to denature proteins. The properties of membranes of membranes also change with temperature. These thermal effects dramatically influence animal fluctuation. Although different species of animals are adapted to different environmental temperatures, each species has an optimal temperature range. Thermoregulating helps keep body temperature within that optimal range, enabling cells to function most effectively even as the external temperature fluctuates.